Good morning to all of you. My name is Shashikant. I am a PhD scholar at Department of Zoology, LNMU. Today I will be talking about my PhD thesis work. The title of my thesis is Regulation of Wind Signaling by E3 ubiquitin ligase Deltex. What is wingless or Wnt? Wingless or Wnt is a paracrine factor. It regulates the wing development in Drosophila. The Wnt name is a fusion of Drosophila segment polarity gene Wingless with the name of one of its vertebrate homologue integrated. You can see that the W comes from the gene Wingless and NT comes from the word integrated. This is how the word Wnt is derived. Wnt and Wingless were initially discovered in different model organisms and were given different names, but they are fundamentally equivalent and share many similarities. There are two loss of function alleles of Wingless: Wingless CX3 and Wingless CX4. What is Deltex? Deltex is a cytoplasmic protein that physically binds to the notch and regulates its signaling activity in a context-dependent manner in Drosophila. There are two mutant alleles of Deltex, a null allele Deltex 152 and a loss of function allele Deltex. Now let's talk about the Wnt signal transduction pathway. On the left side, when Wnt is not present, Frizzled is deactivated. Therefore, the disheveled is also deactivated. GSK3 kinase phosphorylates the beta-catenin and APC polyubiquitinylate the beta-catenin. Therefore, the beta-catenin is tagged for proteasomal destruction. Consequently, the downstream gene is not activated. Now let's have a look on the right side. When the wing binds with the frizzled, dishevel gets activated, and dishevel does not allow GSK3 kinase to phosphorylate the beta-catenin. Therefore, beta-catenin gets transported into the nucleus and it upregulates the downstream genes. How do we study the interaction of genes? There are two ways we can study the interaction of genes. The first is forward genetic approach and the latter is reverse genetic approach. In forward genetic approach, we go from phenotype to genotype. We study which gene is responsible for a phenotype of interest. Whereas in reverse genetic approach, we go from genotype to phenotype. Here, we study function of a particular gene by disrupting it and then observing if it affects the phenotype of interest. Now let's talk about the crossing scheme that I have used during my thesis work. Suppose this female parent is mutant for Deltex gene and this male parent is mutant for Wingless gene. I have used curly balancer in female flies and stubble balancer in male flies. The curly balancer provides a phenotype in which the wing of the fly becomes curved, whereas the stubble balancer provides a phenotype in which the size of the hairs on the thorax region of the fly become very small. When you compare these two pictures with the wild type Drosophila, you can see the difference. The wild type has a very straight and long wing, whereas the curly fly has a curved wing. Similarly, the wild type fly has a very long hairs on the thorax, whereas the stubble fly have short hairs on the thorax region. These are the respective gametes of the female and the male flies. When we combine these two gametes, we get different types or different genotypes of fly. I have written only the required progeny here. The second progeny is very interesting. because here this progeny is heterozygous with respect to Wingless gene and hemizygous with respect to Deltex gene. In this fly, the expression of Wingless has happened in the hemizygous background of Deltex mutant. Now let's talk about the results that I have got. The image B1 shows the wing which is heterozygous with respect to the Wingless gene. The image B2 and B3 shows the wings that are heterozygous with respect to the Wingless gene and hemizygous with respect to the Deltex. In other words, here there is only one healthy copy of Wingless and there is no any healthy copy of Deltex in the background. Therefore, we can see that the wing is not formed properly. There are thickened vein and there are notching at the margin of the wing. Therefore, we can say that reducing the wingless doses in the hemizygous background of Deltex mutants led to the enhanced wing vein thickening and wing margin notching. Loss of Deltex reduces wingless signaling gradient and target gene expression. The image A1 shows the wild type expression of the wingless. Image A2 shows the expression of wingless in the hemizygous background of Deltex mutant. Here we can see clearly that the expression of wingless is reduced in this region. Similarly, the image B1 shows the expression, the wild type expression of sense, and the image B2 shows the expression of sense in the hemizygous background of Deltex mutant. It is clear that the expression of the sense is reduced in this region when we compare this image with the image B1, which is the expression of sense in wild type condition. Deltex modulates the expression of wingless target gene. Overexpression of flag tag Deltex using posterior domain specific engrailed GAL4 results in the complete loss of senseless, cut, and expansion of vestigial in the posterior compartment. The image A1 shows the expression of flag tagged Deltex on the posterior region. The image A2 shows the expression of sense on the anterior region, and there is no expression of sense on the posterior region. The image A3 shows the merged version of image A1 and A2. Similarly, the image B2 shows the expression of cut in the anterior region. However, there is no any expression of cut on the posterior region. Image C3 shows the expression of vestigial on the anterior region, and on the posterior region, the vestigial is expressed in a greater amount. All these images together concludes that whenever the flag tag Deltex is expressed, the expression of sense, cut, and vestigial are reduced. Overexpression of Deltex expands the wingless diffusion gradient. Image A1 shows the expression of GFP in a region where patched GAL4 is active. Image A2 and A3 shows the expression of wingless when in the background the Deltex is overexpressed. The image B2 and B3 shows the magnified version of the image A2 and A3. From image B2 and B3, it is clear that the expression of wingless has enhanced when we increase or when we overexpress the Deltex in the background. Deltex mediated wingless signaling is regulated by endocytosis. Flag tag Deltex co-localizes with RAF5 wingless in the same subcellular compartment. Image A1 shows the expression of wingless in the imaginal disc. Image A2 shows the expression of flag tag Deltex. Image A3 shows the expression of RAF5. And image A4 shows the merged version of image A1, A2 and A3. From the image A4, we can conclude that the expression of flag tag Deltex coincides with the expression of RAF5 and wingless, which means that these three proteins are present in the same subcellular compartment. RAF5 is a small GTPase that is necessary for the endosomal fusion to form early and a more punctated wingless expression was observed when endocytosis is blocked by overexpressing a GDP-bound RAF5 dominant negative with a dorsal ventral boundary specific C96 GAR4. Image C1 and C2 shows that the expression of wingless become punctated when we overexpressed the dominant negative version of RAF5. We know that RAF5 is involved in endocytosis, and overexpressing the dominant negative version of RAF5 lead to the punctated expression of wingless. Therefore, we conclude that Deltex mediated wingless signaling is regulated by endocytosis. DX modulates wing signal transduction. Armadillo strongly reduced in the whole posterior compartment. Image A1 shows the expression of flag-tagged Deltex in the posterior region. Image A2 shows the expression of armadillo in anterior and in posterior region. However, the expression of armadillo in the posterior region is reduced. This is clear from this graph. The intensity of armadillo in the posterior region is less than the intensity of armadillo in the anterior region. Deltex co-localizes with endogenous armadillo. Image C1 shows the expression of flag-tagged Deltex in posterior region. Image C2 shows the expression of armadillo in anterior as well as in posterior region. Image C3 shows the merged version of image C1 and C2. Image C4 shows the magnified version of the image C3. From image C4, it is clear that Deltex and armadillo are localized in the same region. In other words, Deltex co-localizes with endogenous armadillo. Deltex degrades armadillo by proteasome-mediated mechanism. Image A and B shows the immunoprecipitation experiment. In image A, we immunoprecipitated flag-tagged Deltex, and later we immunoblotted armadillo. We get only a single band. In image B, we did the reciprocal of the previous experiment, where we first immunoprecipitated armadillo and later we immunoblotted the Deltex. It again shows the single band. From these two pictures, we can confirm that armadillo interacts with flag-tagged Deltex. Proteasome inhibitor Mg132 treatment resulted in stabilization of armadillo in Deltex overexpressed wing imaginal disc. Image C1 shows the expression of flag-tagged Deltex in the posterior region. Image C2 shows the expression of armadillo in both anterior and in posterior region. However, in this case, the expression of armadillo is same in both the cases, and it can be confirmed from the graph given here. The intensity of armadillo in the posterior region is same as the intensity of armadillo in the anterior region. This happened because we treated this cell with a proteasome inhibitor Mg132. From these data, we conclude that Deltex degrades armadillo by proteasome-mediated mechanism. Proposed model for interaction of Deltex with wingless signaling pathway. This is the known wingless signaling pathway. From our research, we have concluded that Deltex activates the wingless and downregulates the beta-catenin or armadillo. Conclusion: Deltex genetically interacts with wingless pathway component. Loss of Deltex reduces wingless signaling gradient and target gene expression. Deltex modulates the expression of wingless target gene. Overexpression of Deltex expands the wingless diffusion gradient. Deltex mediated wingless signaling is regulated by endocytosis. Deltex modulates wingless signal transduction. Deltex degrades armadillo by a proteasome mediated mechanism. Deltex physically interacts with armadillo. Acknowledgement. My sincere thanks to Professor Spiros Artavanis Sakonas, who teaches at Harvard Medical School, Professor Florencio Serras, who teaches at the University of Barcelona, Professor Hugo Belen, who teaches at Baylor College of Medicine, Professor Yah Nung Jun, who teaches at University of California, San Francisco, Professor Akira Nakamura, who teaches at Institute of Molecular Embryology, Genetics, Kumamoto, Japan, and the Bloomington Stock Center for fly stocks and antibody. Thank you.